Hello there, you're welcome to Biology Lectures with Akeri Loladimiji Philip. So today I'll be taking you through a detailed revision for GCSE Biology Paper 1. And I'll be focusing on the AQA curriculum, okay? And that's the 4 topics you should expect on these are cell biology, organization, infection and response, and bioenergetics. This is called a detailed revision because I'm actually going to go in a bit more detail so that this will help you for your exam. Most of you have about few weeks to your exams. Today is 22nd of April, like your exam starts on 10th. Paper 1 is 10th of May. That's less than 3 weeks from now. So you might want to just sit back and make sure you jot along, or you pause the video if you have to get some stuff in this. All right, so we'll move on to the first part, which is cell biology. So I'm going to start off with what a cell is. So we know that cell is the basic unit of living things, or you can say of life, as it were. All right, now there are 2 types of cell. We have the prokaryotic cell and eukaryotic cell. What you want to remember here is the fact that eukaryotic cells have a membrane-bound organelle, which means they have a true nucleus. All right, that's what it means. So it means their organelles are membrane-bound. Not only that, they also have a true nucleus, as it were. Okay, then the next thing you want to remember is the fact that examples of those are plant cell and animal cell. Please, and also fungus. They actually another guy there, which is protist, but for the sake of your curriculum or specification, the examples of eukaryotic cell you need to know are animal cell, plant cells, and the fungus or fungi plural. Okay, there's other guy there, like I mentioned, the protist is another example, but I won't dwell on that. Now, on the other hand, the prokaryotic cell is those without a true nucleus. So those are the likes of bacteria. Okay, I will try to draw that real quick. But before I do that, so this eukaryotic cell, it has a true nucleus. The nucleus is what that really means is that the DNA is in a structure. There's a nuclear pore, nuclear membrane surrounding it. All right, same thing with this guy. So they are both eukaryotic. But when something is called prokaryotic, let me try to draw something really sketchy here. So that is the I'm trying to draw that. Then I draw something like that. Then I draw that. Now this is the DNA or genetic material of a prokaryotic cell. So now if you notice, this prokaryotic cell, in this case bacterial cell, it's not within a structure. There's no membrane, nothing. So because the genetic material is not membrane bound, it's not in the nucleus, that's why it's called prokaryotic, like false nucleus, as it were. Of course, that's not the only difference you should know between the two of them. You should know the fact that these prokaryotic cells are smaller. Some of the example is bacteria. You should know that some of them have plasmid, which is a second DNA, which helps the bacteria to develop resistance against antibiotics. And you should also know that it has cell wall, like plant cell. Like I told you, it's a detailed revision. I'm trying to touch a lot of things. So pause the video along the line, because that's the only way you can get the best out of this. So you are going to pause the video to understand things and just write things, as it were. Now, before I leave that, I want you to know that for all cells, whether prokaryotes and eukaryotes, all of them have these following stuffs in common. What are those? They all have cell membrane. They all have genetic material. They all have cytoplasm. They all have ribosomes. Now, let me call your attention to something on the screen. Now, I said genetic material. I did not say nucleus. That's because genetic material here simply means DNA, maybe RNA, but just DNA majorly. Now, if I say nucleus, that means I've not put into consideration the prokaryotic cell. When I say genetic material, every one of them has genetic material, whether it is in a nucleus or not. So these four structures, cell membrane, genetic material, cytoplasm, and ribosomes, are common to all cells, whether prokaryotic or eukaryotic. Now, focusing on the eukaryotic cell, in this case, animal and plant cell. They both have nucleus, like we both said. They both have ribosomes, cell membrane, and all that. Now, you should take note of the things that are peculiar to the plant cell. It's a common thing you should know, like it's very common. So you can see the things they have in common, which is the ones they have the two ticks. But pay attention to the fact that plant cells don't have chloroplast, and animal cells don't have chloroplast. Plant cell does. Animal cells don't have permanent vacuole. Plant cell does. Animal cells don't have cell wall, and plant cell does. So this is something you don't forget. Now, please pay attention at this point that you should know the function of each of these structures. Yes. Now, let me quickly take you through that. This function of the nucleus is, whether in animal cell or plant cell, is to control. So it controls the cell. Please, I'm brushing. That's why my writing is very fine from behind, if you get what I mean. So controls the cell activities. Now, sometimes students often say it contains genetic material. Yeah, that's more like description, but it's more, if you really want to say what it does, it is controls the cell activity. Number two, cytoplasm is the site of chemical reaction. The chloroplast is where we have photosynthesis. Why? Because it contains a pigment called chlorophyll. So chlorophyll is in chloroplast. Let me do that again. Chlorophyll is chloroplast. All right. Chlorophyll is just a pigment, while chloroplast is the actual structure. You should know that. Now, the next cell on the list is the cell membrane. So cell membrane just does something simple. It selectively allows, when I say selectively or partially, selectively allows substances in and out of the cell. Wait, what does that even mean? That means it doesn't allow anything to just come in. If you've done osmosis, or when we get to osmosis, you get to understand when it's being defined as partially permeable. So like I said, so being partially permeable simply means it allows some things into the cell and prevents some things from entering, so to say. And of course, it also protects the cell. Just generally protect, because it's the first layer of the cell if it's an animal cell, but second layer of the cell if it's a plant cell, as it were. Okay, so not to get this too rough, let me get this off. The next one to look at is the permanent vacuole. Of course, permanent vacuole contains sap. What is the sap? Basically sugar, or so it just stores. It serves as storage. So in case you're wondering what's the function of the permanent vacuole, is serve as a storage structure in plant cell, as it were. Okay, the mitochondria is the site of aerobic respiration, or you just say respiration for short. Okay, so it's that you just say respiration for short, or you just say aerobic respiration. Okay, then the next thing, ribosome is the site of protein synthesis. You know that. Then the cell wall protects the plant cell. Bear in mind that there's also cell wall in bacterial cell too. So in either way, in both cases, it protects, and it's made from cellulose. And that's something I want you to know now. So the cell wall in plant cell is made from cellulose, while the cell wall in animal cell, or maybe in bacterial cell, is made from something called peptidoglycan, or something called murine. But your emphasis is usually on plant cells. So it's made of cellulose. And what's the function? It keeps the plant cell rigid. If you look at this, so that, and there's something I want to say here. This cellulose cell wall keep the plant cell rigid, and when you put this plant in water, it will not burst. It can suck in the water. It become turgid, which is swollen, but it will not burst because of this cellulose cell wall. Unlike if you were to put this same animal cell in the same water, it's going to like suck in the water by osmosis, then burst, rupture, because it doesn't have cell wall. So if you ask what's the advantage or what's the significance of this cellular cell wall implant, it prevents osmotic pressure or osmotic bursting. Let me write that. It prevents osmotic bursting in the plant, because the plant can suck all the water it can. It wants to, but it's going to burst because cellular cell wall is quite rigid. So that's a quick overview of the functions of the organelle. Let me emphasize here that as far as your curriculum is concerned, your specification is concerned, you don't need to know more than this. Now, if you're doing triple or separate sciences as it's called, you should know that, of course, you should emphasize more on this thing called binary fission, which means ability of bacterial cell to split asexually. Yeah, it's just basically asexual reproduction, whereby whereby the bacterial cell split into two. I did mention plasmid sometimes ago that I said plasmid gives bacterial cell resistance against antibiotics, so to say. So this is why I already explained, talking about the structure of the bacterial cells or prokaryotic cells generally, if you can remember. So that's something you want to take note of. So having established that first part, so that's the difference between prokaryotic cells and eukaryotic cells. So then I'll move on to cell specialization or specialized cells, as it were. So we'll move on to the next part, which is cell specialization. Here, you need to understand that cell specialization actually has, let me say differentiation leads to specialization. So it says when a cell changes, which means they become unique. So if you probably ask what is cell differentiation, it means whereby a cell becomes unique, which is what you're seeing here. I think I want to reduce the thickness of that. It becomes unique. That means it become they change. Yeah, they change. That means they become unique. Then they become specialized, which means they can perform a specific function. Let me give you a quick instance of that. So if I were to draw, just hang on there. Okay, just hang on there with me, just a few seconds. So now this is a sperm cell. I just try to draw there, and this is let me think of, okay, I'm trying to do something real quick. Okay, now this one I drew here is the sieve tube or sieve element in the phloem, and this is a sperm cell. Now the truth is, the sperm cell, as it were, has a tail, a midpiece, a head that contains acrosome and all of that, mitochondria here, that allows it to be, so it is unique because it has a tail. It has a head that has enzyme that can digest the egg, the wall of the egg. It has mitochondria there. So that's so it is different from others. So the process whereby is a normal stem cell change to become this is called differentiation. So after the stem cell has changed, that means become differentiated to become this sperm cell, then this sperm cell becomes specialized for what? Reproduction. So cell specialization refers to a cell becoming specific for its job. So in this case, this cell is only meant to do one thing in the animal's body or mammal's body, which is for reproduction. While this one, the sieve element, or we say the sieve element in the phloem, is adapted to do what? To transport manufactured food, which is what? Sucrose in this case, and amino acid. Bear in mind that what you form on the leaf of the plant is glucose during photosynthesis, but what is being transported is sucrose. So what you expect to find in a phloem or the sieve element of the phloem is sucrose. Please bear that in mind, okay? Hope that made sense. So coming back here, you need to remember these specialized cells and what they do. So I will advise you at this point to actually just pause the video and take note of each of these, the function and adaptation. This is very important. As a matter of fact, most questions always focus more on the adaptations, like what does each of these cells has that makes it to do what it does. That's one major thing you want to know. So what does each of these cells has that makes it to do what it does? So for example, sperm cell does have a streamlined body, a long tail for what? For swimming. This actually is about a 4-marker thing. So let me expand it a little bit. So the streamlined head makes it to move, okay, along the female tract. The long tail it has allows it to swim. Then a lot of mitochondria provide energy, because for it to swim from where it is deposited to where it is meant to be, it needs a lot of energy. So you want to remember the fact that the relevance of the mitochondria in there is for providing energy for the sperm cell to swim. So at this point, I would advise you, please don't pause the video here. All of these are quite explanatory, self-explanatory. Pause the video here. Study each cell, the adaptation, each cell and adaptation as it will. All right. Okay, so we've done this part and this part. So I on purpose would do the practical video separately. Okay. So just know that I didn't forget about this. I skipped because I'm going to talk about that separately. I do have a video on that separately. Okay. Now the next one to look at right now is to look at the structure of the cell a bit more detail. We should know that the cell does have each cell has 23 pairs of chromosomes. Now let me explain what I mean by that. So a normal cell, which means a somatic cell, which means a cell that is not involved in reproduction, has 23 pairs of chromosomes, or you can say 46 chromosomes. So if you say 46 chromosomes or you say 23 pairs, you see saying the same thing. But you should know that if it's a reproductive cell like sperm cell or egg cell or any reproductive cell, is going to be half of what the other cells are. So that means it's going to be 23 chromosomes in each of the cells. It's going to be pairs. That's very important for you to know. Now, you should know this hierarchy. Inside the cell, you have nucleus. In your nucleus, you have chromosomes. Then, if you unwind your chromosomes, you're going to see a structure called DNA. The segments of your DNA is what we call genes, so to say. And the genes controls characteristics, trait, and all of that. I mean, just traits that makes us who we are. Of course, we're going to see more of that in paper 2. So from the biggest to the smallest, like that. But from the smallest to the biggest, like this. Please do know that. So that's like hierarchy of the cell or structures of the cell. Now, you should know that when a cell wants to divide, it needs to do something which is called cell. It undergoes cell cycle. Well, cells normally undergo cell cycle, but I'm going to emphasize something real quick here. Please pay attention to what I'm going to do here. So your cell needs to, of course, replicate its DNA or its genetic material afterwards, then divide. Okay, well, before doing that, there's a stage called mitosis. Mitosis, then divide. What do I mean? So this part where the cell replicates and grows, let me say grow and replicate, this is called interphase. The stage whereby the cell is just growing, then replicating, is called interphase. What does that even mean? Maybe the cell was this size, it has become bigger in size. Now, if it has, say, 46 chromosomes, that 46 chromosome must duplicate and have another 46 chromosomes, because when the cell will split into 2, so that 46 chromosomes can come into each of the cells. This is very important. So this cell replication, this duplication or replication of the genetic material is very important, and this too happens in a stage called interphase, please. Both growth and replication of the DNA happens in mitosis. Having established that, mitosis itself, which is the actual cell division, which we'll talk about later on, is a 4-stage process, which is we have the prophase, the metaphase, the anaphase, and the telophase. Please, I'm rushing a bit so that I won't make the video too long. Okay. Now, having established that part, so I just quickly explained to you. So what happens in mitosis is actually these 4 stages. Now, take note of this. Now, you should take note of the fact that mitosis is in the nucleus. It's a nuclear division. So if you have to define mitosis, you must start by saying that mitosis is a nuclear division that results in the division of cells into 2 identical cells or into 2 daughter cells. So it's a nuclear division. Please don't forget that it happens in the nucleus. It happens in the nucleus. Okay, that's something you want to talk. You should take note of. But the truth is, we still go on to that later on. All right. Now, having established that, let's come to stem cells real quick. Please remember that you should pause the video as time goes on if you're not clear with some things. Okay. Now, having established that, what are stem cells? So stem cells are, well, embryonic stem cells are cells that are undifferentiated, which means they are not unique. They are just, can you see, they are just identical to each other. So this is stem cell. So they've not become unique or specialized. Don't forget, unique means that the cell has assumed a shape. I gave the example of the sperm cell earlier on. So it has become differentiated into something like this, then it becomes specific, which means becomes specialized. So in stem cell simply means undifferentiated cells, which means it has not become unique, and which means it has not become specialized. So it can become anything. Now, the good thing is, it says they have potential of what? Being turned to any kind of cell. So for that reason, they can be used medically to, they have potential of being used medically to treat some diseases like Parkinson's disease, diabetes, and all of that. Of course, like I said, there's a session on this therapeutic use, application of stem cells much later on. Okay, but the truth is, let me just quickly mention that now. It can be used to treat these diseases. However, there's an ethical debate on this, which means that, okay, when you're using embryonic stem cell, that means, you're literally taking a life, or why should you use a cell from an embryo to kill someone else? So that means, that's basically, even though some people argue that, okay, how about if you take the excess embryo formed from IVF, because in IVF, the eggs taken from the mother, after fertilization, you have multiple embryo formed. So why can't you just use that? But people still say that that's a life. Why should you take a life to kill a life? That's something you should know, that you can be asked in your exam, what is the ethical issues associated with embryonic stem cell? The ethical issue is that it's being debated that you shouldn't use life to take care of another life or to kill life, because actually taking the embryo is a potential life, as it were. Okay. Now, on the other hand, we have adult stem cells. Now, they are called stem cell because they can divide, but they are adults because they can't form anything. Let me explain that real quick. Now, this one, let me see, for example, this stem cell can form blood cell, can form sperm cell, can form eggs, can form anything. They can form literally anything. We in A level, they are called totipotent stem cells. They can form anything. However, the adult stem cells can only form some type of cells. For example, in your bone marrow, I hope I got that bone structure. I'm just trying to draw something that looks like a bone. Yeah. In your bone marrow, you do have some stem cells there. Now, those stem cells can only form blood cells. Blood cells like what? Red blood cells, white blood cells, and platelets. So they are restricted to form only certain type of cell. They can't just form other type of cells, as it were. So the common use of adult stem cells are replacing faulty blood cells. Sometimes you can even have bone marrow transplant, making of insulin, making of nerve cells. What that means is, okay, don't forget that these cells can only form the same type of cells. So if you take, there's some cells in the brain that have not yet fully formed. So they can also form nerve cells, but they can't form any other thing. That's the thing I was going to say. So if you take stem cells from the brain, it can only form cells of the brain. If you take stem cells from the skin, they can only form stem cells of the skin. So basically, it just means a cell that's not fully divided, but it can only form that same type of cell from where you have taken it from. That's what adult stem cell refers to. Now, this is very lovely. This is what I was talking about real quick. So stem cells research. So there has been a bit of issues on that. This is what I was telling you. This is the ethical issue. So you want to pause the video here and take note of this. So the for, which means the advantage of that, but against, which means the ethical concern this has raised. Please, you should pause the video here and read that. The next thing you want to know is that plants too have stem cells, and those stem cells are called what? Meristem. Now, one thing you want to know is this, that meristem, which is plant stem cells, are found all through the stages of the plant, all through the life of the plant. But for these ones, for animals, when we get to adulthood, you can find embryonic stem cell. Now, let me say it here. This meristem is equivalent to what we call embryonic stem cell in animals. So that means they can literally form anything, and they have this all through their life. But for us, we only have this. For animals, we only have this embryonic stem cell at the beginning, not all through. That's something you want to know. So these meristems are like some sort of embryonic stem cell in plants, and they are found all through their life, so to say. And that's something you want to take note of. So you might want to pause the video here to further read what is there. Having established that, we move on to this part, which is quickly talking about cell transport, which is the three headings on that: diffusion, osmosis, and active transport. As far as this specification is concerned, you must know these three types of processes by which cell transport things in and out. The first one on the list is diffusion, which is the movement of substances or particles from area of higher concentration towards area of lower concentration. So looking at this here, outside here, we have more of those particles. Now, please pay attention to particles, or either you say particles or you say molecules or substances, but you can't say water or fluid. You can't say water or say solvent. So you should stick to particles or molecules. So if you take here, we have 1, 2, 3, 4, 5. Inside we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 outside. So you expect the substances to diffuse, to move in, and you need this understanding a lot. So in this case here, what allows things to go in and out is your cell membrane, so to say. Now, let's go a bit further. You should know, I made mention of this before, that cell membrane is semi-permeable. Some book will call it partially. Partially is the same thing as saying semi-permeable, which means it does not allow everything to come in. It allows some things to come in, it prevents some things from coming in. That's something you want to know. So it only allows small molecules to come in to get through. For the sake of simplicity, I would define osmosis here. So osmosis is the movement of water particles, or just say water for short, from a dilute solution to a concentrated solution through a partially permeable membrane. Yeah, over the years, like I said, I found that students usually find this a bit confusing. So I will just stick to movement of water particles, or water for short, from a dilute solution to a concentrated solution through a partially permeable membrane. What I'm trying to say is this. Say, for example, I have this is a cell. Then the cell is kept in a container like this. You might want to search up my video on osmosis as well. Then you see, so let's say each of those dots represents water. Each of those circles represents say sugar molecules, as it were. So there, if you that's water there, then let me take some of those sugar molecules out. So according to what we just defined, if you were to take taste the solution inside the cell, and you were to take the solution, taste the solution inside the cell, considering the number of sugars we have, this in this container is going to be sweeter, which means it is what? More concentrated, or it is concentrated, while this is going to be dilute. Now, please pay attention to this. When you say something is more concentrated or dilute, it is always in comparison with something. I'm going to give an example what I mean by that shortly. Now, as far as we are concerned now, this cell is dilute compared to this environment, because the environment has more solutes, which is more sugar. For that reason, we said is the movement of what? Of water from a dilute solution to a concentrated solution. So water will move from inside the cell, will move out, So this cell should shrink because it's going to lose water by osmosis. Now, I'm going to now do something now that when I said that when something is concentrated or dilute is in comparison with another solution, this same cell, now, I'm going to reverse the case now, that we said was dilute. Say, for example, we now take away the sugar in this solution. Now, let's say we just have 1 sugar, 2 sugar, just 3 sugar molecules, for example, please. We can't really actually count sugar in the solution, really. I'm just trying to give it for understanding here. So that means that this inside this, now, let me add more sugar to this cell, has now become concentrated, where this has become diluted. So what we expect now is that water will move from what area of dilute solution to a concentrated solution. In this case, it's going to be from dilute to concentrated, so to say. Bear in mind that another word for dilute is hypotonic, and another word for concentrated is hypertonic. Sorry, okay, let me relax slowly. Hypertonic, then this hypotonic. Okay, so that's something I want you to please take note of in osmosis. I actually just wanted a simpler definition. There are different ways you can define it. Peradventure you have any question with definition you are given at school, just add that in the comment section, or you should contact me from the detail on the screen, then I can always explain to you. But there are about 4 definitions of osmosis, and you know the funny thing, they all mean the same thing, but usually students find them confusing, but they all mean the same thing. Okay, all right. So the next thing I want to look at now is active transport. Now, this is the movement of substances against concentration gradient, which means you are moving things from where they are less to where they are more. For example, we just have 2 molecules in this region A. We have 7 molecules in region B. What we expect normally should be that molecules should move from B to A. But according to active transport, we are going to move against concentration gradient, which means we are going to move from A to B. And for you to force these 2 guys to where you have this 7 guys, you need energy. So if you ask to define active transport, is a movement of substances against concentration gradient using energy. This is very important. You are using energy. You don't want to forget that word. Energy must be in your definition. So active transport is like opposite of diffusion, but you are using energy. This is very important in understanding that. I hope that made sense. So having established that, we move on to gas exchange. Now, gas exchange is both in animals and in humans as well. Okay, so I would hold on. So we look at gas exchange. Yeah, I was trying to actually remove a part which was not needed for you guys now. So you need to understand that we have gas exchange taking place in animals like humans. Now, gas exchange takes place in the lungs. Another word for gas exchange is called ventilation. You don't want to forget that. It's also called ventilation. And so all the structures you have, which is the trachea, which I'm trying to draw there, the two lungs, all of that is to bring the inhaled air to a structure called alveolus, singular, or alveoli, plural. And I want you to please pay attention at this point. So when you have inhaled your air, what happens is this: the oxygen comes in like that and is going to diffuse across the wall of the alveolus. This is alveolus, singular, or some will call it alveolus, either way. So it comes in that way. This oxygen comes in that way, and it goes into the blood. It diffuses across the wall of the alveolus, then into the capillaries surrounding it. At the same time, carbon dioxide, which is CO2, this is oxygen, is diffusing from the blood into the alveolus. So this is going to go out. Now, pay attention to this. The reason why this can happen is because of this. Oxygen, you have more oxygen in the alveolus. So it's going to diffuse from area of what? High concentration in the alveolus to area of what? Low concentration in the blood. So there's low concentration of oxygen in the blood. So because we have more oxygen in the alveolus, it diffuses that way. Conversely, because we have more carbon dioxide in the cell, because the cell has expired, so the cells have released carbon dioxide into the blood. So there are a lot of carbon dioxide in the blood. So we have more carbon dioxide in the blood than you have in the alveolus. For that reason, carbon dioxide will diffuse into the alveolus because we have high concentration of carbon dioxide inside the blood and we have low concentration of carbon dioxide inside the alveolus. This is very important. You have to remember that this gas exchange is happening because of difference in concentration gradients. There's more oxygen in the alveolus than you have in the blood, so it will move down. And we have more concentration of carbon dioxide in the blood that you have in the alveolus, so it's going to move down the concentration gradient. This is very important. You might want to rewind the video if that was not clear enough. So before I leave this part, you should know something very important, that inside there's something that makes the alveolus to do what it does. And what is that? It is that it's the wall is what? It has a large surface area, number one. Number two, it has moist lining, which makes the oxygen or carbon dioxide to quickly diffuse easily or to dissolve. The wall is thin, which means it reduces diffusion distance. So the aim of this is that it reduces diffusion distance. Please don't forget these things. Even though I didn't write them very well, please just take note of what I'm saying verbally. Then it has a good blood supply, which helps to maintain concentration gradient. So if you ask what is the adaptation of the alveolus that makes it to do what it does, these are the basic adaptation. Now, having established that, there's still another structure called villi, which is found in the small intestine, and this villi helps not gas exchange, actually it's actually nutrient exchange. So in your small intestine, what you have there is final digestion and final absorption. I will explain this more in detail in the next session, which is going to be on that should be organization. Yes, you will see how we have a lot of villi. This is what they look like. This is villus, singular. Villus, singular. So two or more are called villi. So these are the guys that increase the surface area of the small intestine to be able to absorb food, as it were. Now, looking at that, that's for us animals. The next thing here is gas exchange quickly in plants. You should know that this is a section of a leaf. This is your upper epidermis. This is the palisade mesophyll. This is your spongy mesophyll. Our focus is on this part, which is spongy mesophyll. Why? Because it has air spaces in between that. Then we have here guard cell. This is guard cell and guard cell. Probably wonder, how do you know that? Because it has chloroplast. Guard cell is the only epidermal cell in the lower epidermis that has chloroplast. That's very important for you to know. So this guard cell controls the space called stoma, singular, stomata plural. So the guard cell is the one that controls the size of the stoma. So the stoma is actually the part of the leaf through which oxygen goes out as a waste product of photosynthesis, and carbon dioxide goes in as a needed product for photosynthesis. Of course, bear in mind that this arrow can be reversed if the plant is carrying out respiration. But either way, you should know that the stoma or stomata is the opening through which gas exchange takes place on the leaf of plants. This is very, very important for you to know. Then also, let me just add this, that this same stoma is the same source through which water leaves the plant through by a process called transpiration. You should know that as well. All right, so move on to the next part, which is topic 2, which is organization. So you should know from your basics that cells form tissues, tissue form organs, different organ forms and organ system, and different organ system working together forms an organism. So at this point, you might just want to pause the video to quickly read through this short definition. Okay, that's something you want to know. So like I said, the practicals I've made videos on this differently, and I don't want to include that in this gross detailed explanation. So I'm just going to move on now to digestive system. Please pay attention at this point. A lot of questions are being asked here, and you need a solid understanding of this to understand the whole thing. So the digestive system is divided into actually two. Into two, we have alimentary canal. Please work with me. Alimentary canal, and we have the accessory organ. You would know why I'm doing this division. So under this alimentary canal, we have this following structure, which I've given an acronym called MOS, LRA. The M is the mouth, the O is the esophagus, the S is the stomach, the second S is the small intestine, the L is the large intestine, and the R is the rectum, and the A is the anus. So all of these ones that are called alimentary canal, I'm going to be circling them now. So the mouth, the esophagus, the stomach, the small intestine, the large intestine, the rectum, and the anus. Please ignore all of these ones. Ignore those ones, okay? Ignore those ones. You don't need to know that. So all those ones I circled are called alimentary canal. Please, what does that even mean? These are structures that food literally moves through in that order. So food goes in this way through the mouth, from the mouth to the esophagus, from the esophagus to the stomach to the small intestine, which is all those parts. Then from small intestine, it goes to the large intestine. From large intestine, it goes through the rectum, from the rectum to the anus outside. Bear in mind that, of course, what came in here, which is the actual food, by the time it gets to this, it has changed into feces. You should know that, okay? But this is called alimentary canal because this is the actual structures. So if you're asked to explain or list out the structure of alimentary canal or mention part of those guys, these are the guys that are called alimentary canal. This is very important. You might want to pause the video here to study this. It's very important you know this, please, okay? Now, I haven't established that, on the other hand, the accessory organs, as far as this curriculum is concerned, or your specialty is concerned, is salivary gland, the pancreas, the gallbladder, and the liver, which is these, or let me use another color for that, So all these guys in blue. So these, gallbladder, and the liver, all those guys are called alimentary canal. Now, so both accessory organs, all those guys are called accessory organ, please. Both accessory organ and alimentary canal form the digestive system. Please, this is very important. So let me quickly take you through the things you need to know about each of this. I mean, you need to know these things. You really need to know these things because this is AQA. You need to know these things. So let me start with the things you really need to know real quick on this. So in the mouth, you have two things happening in the mouth. You have mechanical digestion and you have chemical digestion. Mechanical is happening through the teeth, which means you're physically breaking down the food. And chemical is happening through the enzyme in the what? In the saliva. What's the name of the enzyme? The name of the enzyme is called salivary amylase. What does it do? It converts cooked starch into maltose. That's what you should know there. Okay. Like I said, feel free to pause the video or rewind if you have to. Okay. So that's the function of the mouth. The next thing you want to know is the fact that when you swallow the food, it goes from the mouth to the esophagus. And in the esophagus, what you need to know is that there's no, let me do that here, no digestion here. It's just what transport the food from the mouth to the stomach. And food moves in this process by a process called peristalsis. Peristalsis. Now, caution. Let me write that clearly. Peristalsis. SIS. Peristalsis is not limited only to the esophagus, please. It happens all through the alimentary canal. What I'm saying is it happens all through the structures. So don't think that peristalsis happens only in the esophagus. No, it happens all through the entire alimentary canal. But the first place you encounter peristalsis is this. So peristalsis is just a wave-like contraction of wave-like movement of contraction and relaxation along the alimentary canal. Wave-like movement or wave-like movement of contraction and relaxation, yes, along the alimentary canal. So we are done with mouth and esophagus. The next guy on the list is the stomach. Please pay attention. The stomach, of course, also carries out mechanical digestion. That's becoming too thick. Mechanical by a process called churning. Churning is actual movement whereby the stomach squeezes the food with the gastric juice in it. It also carries out chemical digestion by an enzyme called pepsin. Pepsin is found in the stomach, which breaks down protein into, somebody will say polypeptide or amino acid, so you can say to polypeptide and amino acid. Let me say and amino acid. Okay, so inside your stomach, what you have here is an enzyme called pepsin. So if you're observant, in the mouth, the only thing that was digested was carbohydrates because it contains salivary amylase. In the stomach, the only that was digested, not fully digested anyways, is protein because it contains pepsin. And pepsin is a protease. Protease is any enzyme that digests protein substances or proteinous acid. Okay, so that is that about the stomach. The next thing you want to know is the small intestine. The small intestine is the site of final digestion, okay, and also the site of what? Absorption. This is very important. Now, what does it have to digest? I mean, before I talk about digestion, absorption, let me say something here. In the small intestine, that's why you are supposed to have amino acid. That's why you should have fatty acid, and glycerol. That's why you have glucose. What did I just do? Let me explain what I just did to you. Glucose. Amino acid is the digested form of protein. Glycerol and fatty acid and glycerol is the digested form of fat or lipids. You should know that. While glucose is the digested form of starch. This is very important. So, in your small intestine, will you expect to have these three substances: amino acid, fatty acid, and glycerol, then glucose? This is very important. You might want to rewind the video if you have to do that to make you understand things here. So, amino acid is from protein, glycerol and fatty acid is from fats and lipids, then glucose is from starch or say carbohydrate. And that's what you have in your small intestine. That's what you have. I say is the site of final absorption. While a final digestion, I mean to say, while absorption takes place here is because it contains villi. Now, villi are these structures we saw sometimes ago. Yeah, these ones. This is very important. So you might want to take note of this now, that it increases the surface area of the small intestine. That's number one thing it has. And these villi are one cell thick. So let me say it here that, okay, let me go back to that part. Okay, yeah, there. So villi is one cell thick. It increases surface, or it increases large surface area. Large surface area. It is one cell thick. So it's one cell thick, or you say it is thin, thereby reducing diffusion distance. It has rich blood supply. Yeah. And the surface is also moist. So these are the features that makes the small intestine to be able to do what it does. Now, bear in mind that aside you absorbing amino acid, fatty acid, and glycerol and glucose in the small intestine, you also have water being absorbed. Not totally, but some water being absorbed there. There's also some ions also being absorbed here. That's very important for you to know. The next guy on the list is the large intestine. This is what you have here is what? Undigested food. So whatever did not get absorbed in the small intestine moves to the large intestine. Then one thing you want to know that happens here is there's further absorption of water. So water is further absorbed. Then also we have ions, otherwise called electrolyte, being absorbed, or minerals being absorbed. What do I mean? The likes of sodium is being absorbed, calcium is being absorbed, things like that are also being absorbed in the large intestine. Okay. Then of course, rectum and anus are no, the specification doesn't ask questions on these two really, but just for you to know, the rectum is actually where you have your feces being stored before you expel it out through the anus. Okay, that's very important. Now, that is a quick overview of, well, not just quick overview, that's like what you need for your exam as far as the alimentary canal is concerned. So let me move on to talk about the accessory organs. So move on to talk about the accessory organs. So I will start with, those are the ones I circled in blue. So the accessory organs are these ones. Food doesn't pass through them, but make no mistake, digestion cannot happen without their inputs. For example, the salivary gland secretes saliva. It is inside the saliva you have your salivary amylase, which I mentioned when I was talking about mouth. So this salivary amylase will actually convert your cooked starch into what? Into maltose. Don't forget that. Cooked starch into maltose. So that's the function of saliva or salivary gland there. The next guy on the list is the liver. Now, this one is quite tricky a little. So liver, which is this brown large guy, that's the second largest organ in our body. So this liver does something. It stores bile. Excuse me, it doesn't store. It makes bile from unused amino acid. So it makes bile. Please pay attention to what I just said. It makes the bile. Where this bile is stored where? In the gallbladder. So the function of this one is stores bile while it's synthesized or made. So makes bile. This is usually confusing to students. It makes bile. It makes bile. It makes bile. That's liver. Stores bile. That's gallbladder. So they work together, so to say. So whatever it's made here, it is stored inside the gallbladder. Now, what's the function of bile? That's very important. It's very relevant. So when the food you have eaten comes in from the stomach, this is stomach, it moves to the small intestine, which starts from this point here. In this small intestine, any food that is fatty in nature, if it is fatty food there, it stimulates the gallbladder to secrete the bile onto that fatty food you have eaten. What will you do with the fatty food? It will emulsify, which means it will break it down into smaller droplets. So this is the large fatty food you have eaten. It will emulsify it into smaller droplets, which is to make enzymes that will digest them do their work or job easily. What is the enzyme that digests fatty food? It's called lipase. Lipase, because lipase which works on lipids. So when you've eaten fat or otherwise called lipids, it is emulsified first, which is some sort of mechanical digestion. It breaks this guy down into smaller droplets to increase the surface area for who? Lipase. This is very important. So that's all you need to know about liver and gallbladder put together. Don't forget, this guy makes it, but gallbladder stores it. The next guy on the list is the pancreas. The pancreas has secrete enzymes. It secretes pancreatic amylase, pancreatic lipase, and protease. From the name, amylase work on carbohydrates, majorly starch. Lipase work on lipids, which is what I just mentioned here, or I mentioned here, that was going to work on the emulsified fat. Then protease will work on the protein to break them down into what? Amino acids. This is very important. And that concludes a quick overview of the digestive system. Okay, the next thing on the list is for us to look at enzymes. Well, you should know the fact that enzymes have a way they work. They are called biological catalysts. Why? Because they speed up the rate of chemical reaction, which means that if the reaction was supposed to take, normally without enzymes, food will take days, if not weeks, to digest. But these enzymes has made it easy for us. Makes it easier for food to get digested. But bear in mind, enzymes only work on food only. They work on other metabolic reactions in our body. Now, the thing you want to know here is that enzymes are specific to their substrate. Substrate is what they digest or what they work with. And one funny thing you want to know is that enzymes have an active site that is complementary in shape to what they are going to digest. So every enzyme has an active site complementary in shape to what it's going to digest. This is very important. Can you see that how this clearly fits in? Now, there's something you want to remember, which is called the lock and key model of enzymatic reaction. So the lock is the enzyme. Excuse me, that's not nicely written. The lock is the enzyme, and the key is the substrate. So as you're seeing it here, so this is your lock. It does look like a key anyways, the amount of its key. This is the key. So the reason why that model was being used is to show you that enzymes are basically meant to fit in the way you have your key that can easily fit into a particular lock. That's how they are. But that is based on the lock and key model. You should know that enzymes have a 3D shape, and they have an active site which is specific to a substrate. However, there's another theory called the induced fit model. This induced fit model talks about the fact that when you do have an enzyme, the active site is not totally complementary to the substrate. Rather, when the substrate binds to it, it changes its shape to fit in, or to allow the substrate to fit in. I say that again. The in this fit model explains the fact that the shape, the active site, is not totally complementary to the shape of the substrate. Rather, when the substrate binds, it changes its shape to be able to fit in or accommodate the substrate. That is what the in this fit model refers to, and you need to know that. Then the next thing you want to know is the fact that there are factors that affect enzyme reaction, pH and temperature being the major ones. If the pH is too high, enzyme will become denatured. What does that mean? It is moving from, it's becoming too alkaline in nature. That's not good for your enzyme. If it's going too low, that means it's becoming acidic. It's going to also become denatured. However, there's one particular enzyme called pepsin, the one we talked about earlier on in the stomach. Because of the presence of HCl, this enzyme is the only one that can actually work at a pH of 2. That's quite very low. That's very acidic, but it can still work. But other than that, other enzymes don't work at extreme pHs. They work at more likely alkaline or more neutral pH. Now, on the other hand, temperature, when the temperature is too high, the enzyme becomes denatured. When the temperature is too low, enzymes and the substrate will less likely collide, because what you know is that when you have temperature in a system, according to kinetic theory of matter, the molecules will be moving. So when there's enough temperature in the system, there's more likelihood that the enzymes and the substrate will collide and there will be a reaction. So at a lower temperature, the activation energy will be low, which means there will be less reaction taking place. That's at a lower temperature. But if the temperature is normal, that means there will be normal reaction and all of that. So usually there's this graph I want you to know. This is it. When the temperature is, this is the temperature is going up, reaction is also going up. But it gets to a stage whereby the temperature, this is the temperature here, this temperature on the x-axis, this is the reaction on the y-axis. Now, it gets to a point whereby after this temperature, the reaction begins to drop. Why? Because the reason why it's dropping is because the enzyme becomes denatured, which means the shape, the active site, it loses its active site. The hot temperature or the increased temperature, which will break the bonds of the amino acid that holds it, because your enzyme is a protein. So the protein is made up of amino acids, so the bonds will be broken. So once the bonds are broken, it changes the shape, so the active site will not be the way it was before. So it won't be able to be complementary to the shape of the active site of the substrate anymore. For that reason, the reaction will start dropping, will reduce, will stop literally. So at optimum temperature, which means usually it's always around the temperature of the human body. Our body is around 37, so I think 37 to 30 something, 39, thereabout. I think enzyme reaction has to take place. Beyond that, the enzyme becomes denatured, and for that reason, the reaction starts dropping. That's for you to know about that. Okay. So if you ask to define what denature means, it means when enzyme loses its 3D shape, or when the amino acid starts breaking and the active site changes its shape. For that reason, it can no longer bind to the substrate anymore. These are the examples of enzyme you need to know: amylase, protease, lipase. What they work on is there. Okay, you should remember this. Okay. The next guy on the list is the heart and the blood vessels. So now we look at the heart and the blood vessel. So the first one to look at is the fact that the heart is a large pump which pumps blood around the body. Later on in this section, we're going to look at what the heart looks like. But what do you need to know? You need to know the fact that we have 3 blood vessels in the heart, or that works with the heart: the vein, the artery, and the capillary. Now, looking at this here, you can see that the vein has a large lumen or wide lumen, but it has less elastic wall. So what you want to remember is that the vein has less elastic wall and it has a wide lumen. And what you have in most vein is what? The oxygenated blood. What does that even mean? It means blood that is rich in oxygen. Sorry, rich in carbon dioxide. Rich in carbon dioxide, poor in oxygen. That's what the oxygenated blood means. That's what you should have in your vein. Except one type of vein called pulmonary vein. Pulmonary vein is the only vein that does not carry this kind of blood that is called the oxygenated, rather it carries oxygenated blood. So remember that your vein is wide the lumen is wide in shape. I mean, it's wide and it's less thick. You also know the fact that veins have valves in them which prevent backflow of blood. They also know that veins, you can see IN here, it takes blood into the heart, no exception. All veins take blood into the heart, no exception. On the other hand, arteries have thick elastic wall. What's the reason for that? To be able to handle the recoil. So when the heart pumps blood, it expands and recoil. It expands and recoil. It has a narrow lumen. Yeah, the lumen is narrow. So blood moves at high pressure. It has no valves. And it carries blood away from the heart, or say out of the heart. Don't forget that. And of course, it carries oxygenated blood, which means blood that is rich in oxygen, poor in what? Carbon dioxide. Don't forget this. No valves. However, there's one particular type of artery which is called pulmonary artery, which carries the oxygenated blood. So all arteries except for these ones carry oxygenated blood. Hope that made sense. The next guy on the list is the capillary. This guy is just one cell thick. It has pores. So let me put cells around it. This is where exchange takes place. So say for example, you have glucose here, you have oxygen here. So this glucose and oxygen will pass through to the cell through this pore. And the waste product from the cell, which is carbon dioxide and other stuff, will pass back to the blood. So if you ask that, what's the function of capillary? Capillary is the site of exchange between the cell and the blood. Basically, that's what capillary does. So at this point here, you might want to pause the video here just to be able to read through this information here. Basically, I've explained all of those things already, just for you to get it in full. Okay, the next thing I move on to here is let me talk about the heart first, please. Okay, let me look at the heart here. So the heart has 4 chambers: right atrium, right ventricle. Let me use another color for that red part. Left atrium, left ventricle. Now you should remember this word LORD, Lord, which is the left side of the heart deals with. So L means left, O means oxygenated. So left side of the heart deals with oxygenated blood. That's why this part is made red. While the R and D is right, and D is deoxygenated. So the left side is what? Deoxygenated blood. That's why you can see the fact that this part is blue. Now, pay attention. It is not coincidental that some part of these blood vessels are blue and some are red. So every blood vessel means it is carrying deoxygenated blood. Every red blood vessel means it's carrying what? Oxygenated blood. This is very important. Now, it's very important for you to be to label this. So, as earlier said, this is your right atrium, right ventricle, left atrium, left ventricle. You should know that the thickest part of the heart is this left ventricle. Why? Because the blood from this side is pumped around the whole body. That's something you should know. It needs to generate high pressure to pump blood around the body. But the right ventricle is less thick because it only pumps blood to the lungs. This is the lungs here. This is the lungs here. Okay? That's something you don't want to forget. Now, coming back here, you should know that the heart carries out what we call double circulation, or it's a double pump that carries out double circulation. What are the two that circulation? Primary circulation and systemic circulation. So let me explain each of them to you. I'm explaining the primary circulation now. The blood will move from the right ventricle, RV, will pass through the pulmonary artery, which is this one. This is PA, pulmonary artery. It goes to the left lungs, right lungs. So in the lungs, what does it do? Bear in mind that the right side of the heart is carrying what? Deoxygenated blood, which means blood rich in oxygen. Sorry, rich in carbon dioxide. Okay, so it gets to the lungs. In the alveoli of the lungs, it will do what? Drop carbon dioxide, pick up oxygen. Same thing. It happens at the both lungs simultaneously. Drop carbon dioxide, pick up oxygen. So then at this point, the blood becomes oxygenated. So it returns back to the heart through pulmonary vein. Through pulmonary vein. Okay? So the pulmonary vein brings oxygenated blood to where? To the left atrium. And that marks the end of pulmonary circulation. So I run through that real quick. From the right ventricle through the pulmonary artery to the lungs, gas exchange takes place in the lungs. It comes back to the left atrium through pulmonary vein. Okay, that's what you want to know there. On the other hand, I think I should use another color now. Systemic circulation will start from here, the left ventricle. So blood is pumped from the left ventricle through the aorta around the whole body. I think that color is not good enough. I should use black. So from the left ventricle through the aorta to the upper part of the body, part of the body. So it goes like that around the body. What is it doing? It is dropping the oxygen, picking up carbon dioxide, dropping oxygen to the cell, picking carbon dioxide. It does that around the whole body and comes back to the heart by two vessels called superior vena cava. This one from here, this is upper one. Superior means up. And the other one here called inferior vena cava. Okay, so it brings the blood to where? Right atrium. And that is the end of systemic circulation. So I run through that again from the left ventricle through the aorta, from the aorta around to the whole body, upper part of the body, just around the whole body. Then it comes back through the vena cava, inferior and superior vena cava, to where? The right atrium. And that explains that. You might want to rewind the video here if that was too fast. Okay. And that explains how double circulation works. The next thing you want to know is that the heart has some electrical structure that provides, they are called pacemaker, that provides electrical impulses that makes the heart muscles to contract and relax. And we have two major of those. We have the SA node, which is called sinoatrial node, and the AV node. So this one sends the impulse to the wall of the heart. That's why the heart muscle contracts and relaxes. The whole time, even from the fetal, from before we were born, the SA node inside the heart has been generating electrical impulse, which has been supplying the wall of the heart, providing that's why the heart is called myogenic. But you don't need to know that word myogenic now, but just know that the heart has electrical structure called SA node and AV node, which allows it to be able to contract and relax all the way. So the emphasis here is that pacemaker is electrical generating structures inside the heart, and if something goes wrong with these guys, you will need to change it with, replace it with an artificial pacemaker. Now, before I leave that, it just crossed my mind that I have not explained something. So these are valves in the heart. So the function of the valves in the heart is to prevent backflow of blood. That's all you need to know. The valves in the heart, which separate the right atrium from the right ventricle, from the left atrium from left ventricle, these valves have name, but you don't need to know the names now. It is to prevent backflow of blood. So when blood moves here, our blood wants to move from this place to the next place, this guy shuts close, so to say. Now, before I leave this part, your SA node is found in your right atrium. That's a very relevant information you don't want to forget. It's on the right atrium. Okay, hope that made sense so far, so good. So here is the artificial pacemaker I mentioned. So in case the natural SA node and AV node is not working properly, then this is when we have the electrical, this is made from lithium batteries, so it can stay for 5 years, or I don't know, it depends on how it is made. So it will be supplying the heart with electrical signal for it to contract. The next guy on the list is the coronary heart disease, short form CHD. So CHD is usually when there's a blockage in the coronary artery. Coronary artery is those blood vessels that supplies the wall of the heart. They supply the wall of the heart. I think I have a video on that. You can check my channel on that. So this coronary artery, it's a normal artery, as you know. But if it happens that there's accumulation of cholesterol in there and it goes so bad that it blocks the entire artery, when it blocks the artery, that means whatever this artery supplies around here, the blood will just start flowing backwards. So this part of the heart will not get enough blood supply, and eventually that part of the heart will die. So for example, this is the part that's being supplied. So when this guy doesn't get enough blood supply, that means there's no oxygen, no glucose, and these two are very important for the heart muscle to contract. So if these two are not getting to the heart muscle, that part of the heart muscle would end up dying, and one particular fatal day or bad day, that part of the heart will shut down and the person may end up having heart attack. So emphasis, it is because there's a blockage in the coronary artery. How does it happen? Because of fatty deposit. That deposit is called cholesterol. Okay, and it can actually lead to death if it's not managed. So the main symptoms is people have chest pain. Then chest pain is just at the beginning. Then heart attack or heart failure will be when the heart shut down, when there's total blockage, as it were. So the things that increases the likelihood of these things is the diet, eating too much fatty food, and less less fruity vegetables is not good for people. Smoking and stress. These three have been found to be a major causative agent that can cause CHD. So you might want to pause the video at this point to read these stuffs, okay, to yourself. Now, how is this being treated? I think I need to show you that real quick. If it's not mentioned yet, let me say that real quick. So this CHD is being treated using two stuffs, a drug called statin. Statin will reduce the level of cholesterol, and another instrument called stent. Stent will help to widen. It's like a mesh, wire mesh. We widen the wall of the blood vessel, like the part that has been, for example, where I used, here. Imagine here where I have the blood vessel already being blocked here. So you put stent here, so stent will expand it. Okay, I think I also have a video on that or not, but just know stent is a metallic structure put into the wall of the coronary artery to expand it since it has been blocked already. Okay, so these are the two treatments you have for CHD. Okay,
blood has two components: the cellular component, which is the red blood cell, white blood cells, and the platelets. Then the non-cellular component, which is the plasma. So if you actually take out time to read, that's what you're going to see. Now, quickly, it's very important for you to remember some few stuff here. The function of the red blood cell is to transport oxygen and carbon dioxide. Yeah, and sadly carbon monoxide, CO, but you should know this, that's what it transports. The white blood cells is to protect us against infection, while platelet is for blood clotting. That means when you have a clot, this is your blood vessel and it's a blood to flow out, it forms some blockage on that, so it prevents bleeding for the under normal circumstances. Then the plasma, this one is where you have your glucose dissolved, your amino acid dissolved, all sort of nutrients. So the point is, if you ask for the function of plasma, that's why you have nutrients being transported, which includes glucose, amino, all sort of food substances, even waste as well anyways. So not just plasma, not just the nutrient, not just nutrients, even the waste product also is being transported in there. Okay, so you can just pause the video here to further read this. But one thing I want to point out is this: carbon dioxide is being transported both in plasma and also in the red blood cell. So about 75% of carbon dioxide is transported in the with the red blood cell, and about 25% is transported in the plasma. Okay, so that's something you should know, because sometimes student gets confused. Okay, I saw carbon dioxide being transported also in the plasma. Yes, so both red blood cell and plasma transport carbon dioxide. Okay, you can pause the video here. So virtually everything here that was written here has been explained. The next kind of thing you want to focus on is cancer. Cancer basically refers to uncontrolled growth and division. So when we say uncontrolled, that means the cells just keep dividing and dividing. Now, one thing you want to know is that this word, when the cell divides uncontrollably, it becomes a mass, and that mass is called tumor. Now, that tumor can be in two ways. That tumor can be in a capsule or within a membrane. That's called benign. But if that tumor, after accumulating to mass of cell, then starts spreading through bloodstream to other organs, then that becomes malignant tumor. That is very important. So a tumor that spreads is called malignant tumor. However, the process of spreading itself is called metastasis. So when the tumor cell spreads to other part of the body, that is called metastasis. All right. Now there's a quite difference in the way they manifest. For example, the benign tumor is usually very slow. It's within a membrane, like I said that, and usually when you take them out surgically, they don't grow back. Now, generally, they don't spread around the body, and they are less life-threatening. However, if they are found in a particular organ, that is, if you take them out on time, they won't affect you so much. But the truth is that they will end up affecting the organ where they are found. Even though they are less life-threatening, they can also, generally they are less life-threatening, really. But if they are found in, say, in the brain, they can easily affect the person, or in the lungs. Okay, but generally speaking, if you take them out, they are less life-threatening, but they can actually affect the organ in which they are growing in generally. On the other hand, malignant tumor that spreads are cancerous, which means that they can spread around and also affect other parts. That's usually it grows fast, it goes through bloodstream. That's what I just explained here earlier on, and it can break, so to say. So you may want to pause the video here just to see the differences between malignant tumor and benign tumor. Remember, cancer just means that the cell is reproducing rapidly and it forms a mass called tumor. Having established that, we quickly take a look at plant tissue, organ, and system. You should know that plant has tissues itself, and that tissue are massed to form something called leaves. So that means leaves is an organ. What other organ does plant have? Flower and fruits are other organs present in plants. Okay, now focusing on leaf as the specification demands, leaf is a site of photosynthesis. Why? Because it contains chloroplast, an organelle which contains chlorophyll, a pigment. I said that before in one of the other parts, that the pigment is the chlorophyll that absorbs sunlight, but the organ, or the organelle, I beg your pardon, is the chloroplast. So you have chloroplast look like that
that has structures there like that, then it has pigment. So those pigments are called chlorophyll, and that's the actual guy that absorbs the sunlight. Okay, so this is your photosynthetic word equation. Okay, and that's something I think you should know. So what does the plant has? I mean, what does the leaf has that makes it to do what it does? You might want to read this now, but I'm going to summarize it real quick. It is quite wide, because normally leaves are always wide in shape, and that is to increase the surface area to volume ratio. Then it's always thin. Why? To reduce the diffusion distance for oxygen and carbon dioxide to go in and out. Carbon dioxide will come in, and oxygen will go out as a waste product. So that's to reduce diffusion distance. Also, within your leaves, we have something called vascular bundle, which is made up of two things: your xylem and phloem. What xylem transport? It transports dissolved minerals and water from the soil, while your phloem transports manufactured food, which is your glucose or your sugar or your sucrose, as the case may be. That's something you don't forget. So now you need this understanding of the leaf to make this make more sense. So looking at this here, I will run through this real quick. So, well, most of the things I've mentioned here, but I'm just going to emphasize some few stuff here. So looking at your leaf here, the first layer is always this waxy cuticle, which usually just prevent or protect the leaf against drought, or let me say harsh weather condition, or sunlight, or UV ray. Okay, the next guy on the list is the epidermis, which also protect the plant, but it does something. It is transparent enough to allow the sunlight to pass through. Okay, that's something you want to remember. Now, I just said that the upper epidermis allow the sunlight to pass through. Okay, then to pass through to what? To palisade mesophil, which is the actual major site of what? Photosynthesis. Why? Because it is made up of a lot of chloroplast, number one. Number two, this layer has the highest rate of photosynthesis because the cells are clustered. They are arranged in clusters. Okay. The number three is that they are arranged perpendicular, which means 90 degrees. I mean, 90 degrees to the surface. Yeah, I mean, that's what I mean to say. So it is arranged perpendicular to the surface of the leaf. So three components makes this layer more active in photosynthesis, which means it has more chloroplast. Okay, it has the cells are arranged in clusters. Okay, then also they are arranged perpendicular to the surface of the leaf. Okay, the next guy on the list is mesophil. This guy also has chloroplast in it. However, the only thing you want to remember is the fact that it has spaces for air. Another thing I want to remember is that it also serves as a temporary storage of what? Of starch, or say of manufactured food. Yes. So it serves as a temporary storage of manufactured food. That's aside the fact that it has the airspace. And what's the function of the airspace? For gas exchange, for carbon dioxide to go in during photosynthesis, and oxygen to come out. Bear in mind that this going out and coming in is controlled by a cell called guard cell, and that guard cell is being shaped, and it has a hole in between them, which is called stomata plural. So guard cell is the being shaped structure. The space in between that is called stoma, singular, stomata plural. Oh, that made sense. So then, like I said earlier on, you have your xylem and your phloem. So this part is your xylem, and this part is your phloem. So like I said, so can you see how it works? So you have stored the manufactured food temporarily in the space there, then it is transported by who? By the phloem to other parts of the plant. But amazingly, your xylem brings water plus dissolved minerals like magnesium and the likes to this part of the plant for photosynthesis to take place. Because remember that your photosynthesis is what? Carbon dioxide plus water. So this water must have come or came from the soil through the xylem. You might want to rewind the video at this point if it's too fast, so to say. But basically everything I've explained is in there if you want to pause that. Okay.